Hello, my name is Erica Huey.
Hi, my name is Senami Saka-Shenayon.
My name is Wilfred and today our group presentation is sex determination in plants.
And some topics that we're going to cover today is the developmental process of sex determination in plants, the biological significance of sex determination in plants, and sex determination at the mechanistic level. So, starting off with the developmental process of sex determination in plants, we want to know how do plants develop their sex. So, sex determination in plants is the process that controls whether a plant will develop male, female, or both reproductive organs. Different plants have different reproductive strategies. So, this includes for dioecious plants, they have separate male and female parts. And then for monoecious plants, they have both male and female parts in one plant. As far as gene control of sex determination in plants, in dioecious plants, sex determination genes are located in regions of sex chromosomes that do not recombine. For example, in the white companion, the Y chromosome determines sex by suppressing the female traits, and the suppression of female traits leads to the promotion of male traits. In monoecious plants, sex determination is controlled by regulatory genes on autosomes, and these genes influence whether male parts or female parts develop fully in each plant. So, for example, in melons and cucumbers, a gene called CMWIP1 decides that sex fate. And hormonal influences on plant sex. Um, there's sex determination loci that have been involved in hormone biosynthesis pathways, and the hormones that are involved in that include ethylene, GA, and JI. And for JA in maize, this inhibits stamen development in ears, and then there's also no effects on pistil development. Whereas in ethylene in cucumbers, it promotes pistil development and inhibits stamen development.
Now we're going to move on to the next topic, which is the biological significance of sex determination in plants. Plants are biological. Sex development in plants is essential for ensuring reproductive success. It helps plants to determine whether they would require male or female reproductive organs. For instance, in the hermaphroditic plants, they benefit from self-pollination and cross-pollination, ensuring higher reproductive efficiency. For instance, in monoecious plants, they separate male and female flowers on the same plant, which reduces inbreeding while ensuring pollen transfer. And then dioecious plants, they force out crossing, which increases genetic variability and resilience. Another biological significance of sex determination in plants is that it facilitates adaptive flexibility. This allows plants to alter their sex expression based on environmental conditions, which can maximize reproductive success in fluctuating environments, such as temperature-dependent sex determination, which allows plants to adjust reproductive strategies in response to seasonal changes, and also light influence on sex expression in plants like spinach, which ensure reproductive timings align with favorable growing conditions. Another Another biological significance of determination in plants is that it helps with enhancing genetic diversity and efficient allocation of resources. So, for example, female reproductive structures tend to require more energy investment, and male tend to require less resource intensive. So therefore, plants in high nutrient environments tend to develop more female flowers to maximize reproduction, and plants in stressful environments produce more male reproductive structures to favor the environmental conditions. And then this also helps with evolutionary fitness, which reduces the risk of harmful genetic mutations accumulating in populations, and it helps with to increase genetic diversity in populations, which makes them less susceptible to widespread infections. Lastly, Sex determination in plants helps with agriculture and horticultural applications. Understanding sex determination in plants helps farmers to select for traits that they want in certain plants. And farmers can use hormones like GA or ethylene inhibitors to manipulate sex expression in crops. This also allows them to increase fruit yield. And lastly, in commercial cannabis cultivation, female plants are preferred because they produce high levels of cannabinoids. And this allows for farmers to help eliminate undesired male plants early.
And lastly, is sex determination at a mechanistic level. Sex determination in plants begin at a genetic level. Some plants use sex chromosomes like XY, ZW, or exosystem to define male or female identity. Others don't rely on full chromosomes, but rather on specific sex determining genes, such as the CMM1 gene in kiwi fruit. By having different sex chromosomes can create an imbalance in gene expression, which plants correct using dosage compensation. This ensures both sexes develop properly despite genetic difference. However, genes alone don't determine everything. Gene expression can be fine-tuned through epigenetic modification. Epigenetic mechanisms act like switches, modifying gene activity without changing DNA sequences. Three major players influencing sex determination. Firstly, DNA methylation adds the methyl groups to the DNA, turning genes on and off. For example, in permism, methylation at the OGI locus determines whether a plant develops as female or male. Secondly, histone modification change how tightly DNA is packed, affecting gene accessibility, regulating gene-specific insects, and expressing genes based on developmental cues. Lastly, small RNAs regulate gene silencing, fine-tuning sexual identity, and controlling reproductive traits. However, beyond genetic and epigenetic controls, hormones provide a biochemical signal that drives sex differentiation. We have GA pathway. GA triggers DELA protein degradations, allowing activation of male promoting genes like TDF1, meaning GA acts as a molecular switch favoring male traits. Ethylene and cytokinin pathway. Ethylene binds to ETR1, activating EN2 and EN3, which promote female organ development in plants like melons. Cytokinins also regulate floral identity, balancing male and female traits. However, hormonal pathways don't work in isolation. They are influenced by environmental factors. So lastly, we have environmental factors like light, temperature, and nutrition. Light regulation, photoreceptor control the constant gene expression, influencing flowering and sex differentiation based on date length. Temperature sensitivity, high temperatures alter SBL and ZZ gene expression, which regulates reproductive development. This means climate condition can shift plant reproductive strategies. And lastly, nutrient hormone crosstalk. Nitrogen availability affects hormonal balance, altering GA and ethylene levels, which in turn influence sex expression. And the next slide is our references for the whole presentation.
Understanding these developmental, biological, and mechanistic processes not only helps us grasp plant development, but it also aids in agriculture, breeding, and biotechnology. With this foundation, we can explore ways to manipulate plant reproductive systems for crop involvement. Thank you.
Thank you for listening.
Thank you for listening.